Hello, I'm David Eveleth, and I'll be presenting today on the accelerated healing of corneal endothelial lesions induced by an engineered fibroblast growth factor. Here are our financial disclosures. This research was funded in part by the National Institutes of Health under the NCATS TR&D program. Fuchs endothelial corneal dystrophy is the most common reason for corneal transplantation, with tens of thousands of transplants done every year. Typically, the surgeon excises the entire 8 to 10 millimeter diameter Decimus membrane on the inner surface of the cornea and replaces that with donor tissue. An alternative procedure, known as Decimus stripping only, or DSO, is currently being explored. In this procedure, a much smaller area of only the central Decimus membrane is removed, leaving the peripheral endothelium intact. In many, but not all patients, the peripheral endothelium then migrates centrally to restore endothelial function. The advantages of DSO are that it's a much simpler surgery, it does not require immunosuppression, and it does not require donor tissue, which is in short supply. The disadvantages of DSO are that visual recovery is much slower than transplantation, and a significant fraction of patients will not recover and will then need a transplant. If the rate of visual recovery from DSO could be accelerated and the success rate increased, DSO would become an attractive choice for many patients and would remove the need for donor tissue that is in short supply. Therefore, we looked at what controls the migration and proliferation of corneal endothelial cells, and in fact, it was already demonstrated for many years that fibroblast growth factor can accelerate regeneration of the corneal endothelium after surgical wounding, and that FGFs were potent stimulators of both migration and proliferation of corneal endothelial cells, or CECs. We asked if FGFs could be used in this setting. Unfortunately, there are good reasons why FGF has not been developed as a drug. FGF1 and 2 were first described as growth factors in many tissues, including the eye, but they're quite unstable in biological environments. FGF1 has been engineered by nature to signal over short spans of time and distance, and one of the ways nature did this was to make this molecule very unstable. It is subject to unfolding and then proteolysis, and it is also subject to oxidation at the 3 free cysteine residues, the oxidation of any one of which will inactivate the protein. So we took the approach of substituting out 2 of the 3 free cysteine residues and converting the third to a disulfide bond that prevents the unfolding of the protein. This results in a molecule that is much more stable and somewhat more potent than the native FGF1. To model endothelial damage, we first used a rabbit corneal organ culture model that is a modification of the method developed in Alex Lujamov's lab. Corneas were placed in a hemispherical well without media, and a lesion in the endothelial layer created by laying a 4 millimeter circle of filter paper saturated with N-octanol on the endothelium for 30 seconds. This creates a circular lesion that can be stained using trypan blue. The corneas are then placed into suspension culture to recover. The size of the lesion can be followed by repetitive trypan blue staining and quantified using image J. In this model, lesioned corneas will recover over time. The initial lesion is generally 30 to 40% of the total corneal area, and the area of trypan blue staining can be expressed as a percent of the corneal area, and it will decrease over time with complete recovery at about 7 to 10 days. Incubation of these corneas with engineered FGF1s accelerates this process, with healing of a little bit less than 30% of the lesion area in a control cornea versus about 45% of the lesion area in FGF-treated corneas at 3 days. The proposed route of administration of an FGF would be by intracameral injection at the time of the DSO surgery, and that would not result in the exposure of the CECs to drug continuously. It would result in a short-term exposure. Therefore, we simulated what would happen with this method of administration by exposing the corneas after lesioning to TTHX1114 for just 1 hour or for 1 day, and comparing that to continuous exposure to drug for 3 days. All measurements in this experiment are done after 3 days of culture, and the magnitude of the effect is similar for each duration of exposure. This suggests that administration at the time of surgery may be effective. We then moved to a human organ culture model because human corneas are a more relevant model Rabbit CECs proliferate more easily than human CECs, and the rabbit is considered to be a permissive model as compared to human. In this case, we performed the DSO using a full decimate stripping rather than a chemical lesion. So not only are the endothelial cells removed, but the underlying decimate's membrane is also removed. Pairs of corneas from the same donor were used to control inter-individual variability in recovery. In this human organ culture model, the recovery from the lesion is much slower than in the rabbit model. Treatment with TTHX1114 does accelerate recovery, with less than 30% of the lesion area recovering at 14 days in the control group versus more than 80% in the treated group. And this experiment was done with 3 different pairs of corneas. An important aspect of the recovery is the issue of the reformation of a functional endothelial layer. CECs can be visualized using alizarin red staining, which stains the cell-cell junctions. In both the control and drug-treated corneas at 14 days, a normal endothelium with a hexagonal array of CECs can be seen outside the lesion. In the lesion area, where CECs have migrated in and reformed a monolayer, the array is disordered. It will be important to see if the monolayer develops the normal hexagonal array over time, but maintaining these corneas in organ culture for long periods of time is challenging. In summary, this engineered FGF1, TTHX1114, accelerates the healing of corneal endothelial lesions in both the rabbit and human organ culture models, and so this drug may be a useful adjunct to decimate stripping only in Fuchs patients. Trefoil would like to thank the Lions Eye Institute for supplying corneas and all of our collaborators and contributors and our funding sources.